Hello students, in this video I am going to explain how to solve some questions related to meiosis. Okay, this is the question. If an egg of an organism has 10 pg of DNA, 10 pg is nothing but picograms. 10 picograms of DNA in its nucleus, that means egg nucleus is with 10 pg of DNA, amount of DNA present in the nucleus of egg. Egg is haploid, is 10 pg. Okay, now we have to find how much DNA would be present in the diploid cell of the same organism during different phases of meiosis, all these different phases of meiosis. Okay, so egg is actually haploid. So what we need to find? We need to find the amount of DNA during different phases of meiosis. Okay, so egg is haploid. Now this is G1 phase. G1 phase is nothing but meiocyte enters into G1 phase. Meiocyte is diploid. Meiocyte is nothing but the cell that undergoes meiosis is called as meiocyte. The cell that undergoes mitosis is called as mitocyte. Now coming to meiocyte. So meiocyte is diploid. So when this meiocyte is diploid, what will be the amount of DNA when it enters into G1? 20 pg. Amount of DNA will be 20 pg. Amount of DNA will be 20 pg. So what is given? Amount of DNA in the egg cell is given. That means haploid gamete is given. We need to find the amount of DNA in different phases of meiosis. In its diploid cell. That means in its meiocyte. Next coming to G2. So what happens? Amount of DNA gets doubled during S phase because of the replication of DNA. So during S phase, this 20 pg becomes 40 pg. 40 pg. During S phase. G2 phase also same. And prophase 1 also same amount of DNA. Next, anaphase 1 also entire cell same amount of DNA. In each daughter cell of meiosis 1, 20 pg. Because during anaphase 1, what happens? Half of the chromosomes move towards one pole and half of the chromosomes move towards another pole. So here half of the chromosomes are moving towards one pole of the cell in the form of 20 pg DNA, and half of the cells are moving towards another pole in the form of 20 pg DNA. So that each pole becomes one daughter cell. So in each daughter cell of meiosis, which are formed after meiosis, is with 20 pg of DNA, amount of DNA. Next coming to anaphase 2. So daughter cells of meiosis 1 enters into meiosis 2. Anaphase 2. In anaphase 2, the amount of DNA will be same. Entire cell we need to consider. At each pole of anaphase 2 means we need to consider 10 pg amount of DNA, because half of the DNA, half of the chromosomes in the form of half of the DNA in the form of chromosomes move towards one pole and half of the chromosomes move towards another pole. But in the entire cell, same amount of DNA, 20 pg. At each pole of telophase 2. At each pole of telophase 2 is nothing but each pole of anaphase 2. That is 10 pg. 10 pg. So at each pole of telophase 2 becomes what? Each daughter cell. So in each daughter cell, how much? 10 pg. That daughter cell is nothing but gamete. That is egg. So how much amount of DNA is there? 10 pg amount of DNA is there. Okay, for the same question, let's find the number of chromosomes present in during different phases of meiosis. Okay, so now this egg is with let's say 10 chromosomes. 10 chromosomes. So now let's find the number of chromosomes and chromatids present during different phases of the meiosis. Different phases of the meiosis, right? Okay, number of chromosomes now. Actually in egg, how many chromosomes are there? 10 chromosomes are there. Actually egg is haploid. Now the cell that enters into meiosis is diploid. That is meiocyte is diploid. So number of chromosomes, how many will be there? 20 chromosomes will be there. And each chromosome during G1 phase is represented by how many chromatin fibers? 1 chromatin fiber. So how many chromatids? 20 chromatids, because number of chromatin fibers are nothing but number of chromatin fibers is equals to number of chromatids. So number of chromatids. These are number of chromosomes. Chromosomes, right? Okay, now G2 phase. So what happens during G2 phase? Amount of DNA gets doubled, number of chromatids also gets doubled, but the number of chromosomes remains same. We already studied. So same configuration in G2 also, prophase 1 also, anaphase 1 also. Anaphase 1. So same thing, there will be no change with respect to number of chromosomes and chromatids. Now in each daughter cell, in one daughter cell, half of the chromosomes will be distributed to one daughter cell and half of the chromosomes to another daughter cell. So number of chromosomes in each daughter cell after meiosis 1 is 10, and each chromosome is with how many chromatids? 2 chromatids, because there is separation of homologous chromosomes where each chromosome is with 2 chromatids. Hence, as there are 10 chromosomes, number of chromatids is 20, as each chromosome is with 2 chromatids. Next, coming to anaphase 2. So this daughter cell only enters into meiosis 2, which enters into prophase 2, then metaphase 2, then into anaphase 2. So what happens due to splitting of centromere? Number of chromosomes gets doubled. So number of chromosomes gets doubled. So here how many chromosomes? 20 chromosomes. Number of chromatids? 20 only, because in this phase, during anaphase 2, number of chromosomes gets doubled, but the number of chromatids remains same. So chromosomes are getting doubled, but chromatids remain same, because each chromosome is with 1 chromatid due to splitting of centromere. Next, coming to at each pole of telophase 2. Each pole of telophase 2 is nothing but each daughter cell. So to each daughter cell, 10 chromosomes are distributed to one daughter cell and 10 chromosomes to another daughter cell from each parent cell. So number of chromatids will be 10 in each daughter cell of meiosis 2, or at each pole of telophase 2. So this is about how to solve the questions related to meiosis, and I'll give you some more questions. Let's see some more questions related to meiosis. Okay, another question is, find the number of meiotic divisions required for the formation of of 100 seeds. 100 seeds or grains or zygotes. Okay, so to solve this type of question, that is to find the number of meiotic divisions required for the formation of seeds or grains or zygotes, we have to use the formula n plus n by 4. N plus n by 4, where n is the number of meiotic divisions required for the formation of female gametes, that is egg. Number of meiotic divisions required for the formation of female gametes. Female gametes or eggs. Okay. N by 4 is nothing but number of meiotic divisions required for the formation of male gametes. When male and female gametes fuse together, what will be formed? Zygote, that develops into embryo of the seed or a grain. Okay. So N by 4 is nothing but number of meiotic divisions required for the formation of male gametes. So what we can take now, 100. N is nothing but number of meiotic divisions required for the formation of female gametes. So 100 plus 100 by 4. This is for female gametes, this is for male gametes. So 25. So answer is 125. So how many meiotic divisions are required? 125 meiotic divisions are required. So for the same question, find the number of meiotic spindle apparati required for the formation of, we solved meiotic divisions now. Now let's see meiotic spindle apparati required for the formation of 100 seeds. Spindle apparati required for the formation of 100 seeds. Okay, for this what we have to use same formula to find the number of meiotic divisions. After finding the number of meiotic divisions, we have to multiply the number of meiotic divisions with 3, because one meiosis, per one meiosis 3 spindle apparati will be formed. Per one mitosis 1 spindle apparatus will be formed. So here how many meiotic divisions? 125. So 125 meiotic divisions into 3, because per one meiosis 3 spindle apparati. So how many? 375. 375 meiotic spindle apparati are required for the formation of 100 seeds or 100 grains or 100 zygotes. Okay, another question I will explain you. So now the question is, find the number of meiotic divisions required for the formation of like 120 pollen grains or 100 pollen grains, let's say 100 pollen grains. Find the number of meiotic divisions required for the formation of 100 pollen grains. Pollen grains. Where from pollen grains will be formed? From microspore mother cells. For solving this type of problem, to find the number of meiotic divisions required for the formation of pollen grains, we have to use the formula n/4. I'll explain why we have to use this n/4. Okay. So actually a microspore mother cell present in microsporangium of the anther, microspore mother cell present in microsporangium of the anther undergoes one meiosis, undergoes one meiotic division, resulting in the formation of 4 microspores. These 4 microspores only develop into 4 pollen grains. That means one meiosis results in the formation of, one meiotic division results in the formation of 4 pollen grains. So we have to take n/4, where n equals to number of pollen grains, 100/4. So here n equal to number of pollen grains. Number of pollen grains. So 100/4. 25 meiotic divisions. So for the same question, meiotic spindle apparati means 25 into 3, because per one meiosis how many spindle apparati? 3. So 25 into 3, 75 spindle apparati, meiotic spindle apparati. Meiotic spindle apparati. Okay, like this we need to solve questions. So hope you understood all these concepts. So in this chapter I started with what is cell cycle, what is generation time, and I explained a question related to, numerical type of question related to generation time. And then we started with interphase cell cycle, which consists of interphase and M phase. In interphase I explained about G1, S and G2 phases. Then we started with mitosis. We completed mitosis and some tricks to solve mitosis type of questions I gave, and then we studied about the significance of mitosis. Then we started with meiosis. Meiosis 1 we completed, then meiosis 2 we completed. In between meiosis 1 and 2, interkinesis exists. That interkinesis also I explained in detail. So, and I gave some tricks to solve questions related to meiosis also. So this is all about cell cycle and cell division. In the next class, in the next video, I will be starting with a new chapter. Thank you, students.